Hello, thank you for watching this video as part of BioDigital 2020. My name's Katie Maxfield and I'm with DNASTAR. For those of you who don't know, DNASTAR is a software company. We're headquartered in Madison, Wisconsin, and we create comprehensive software for both sequence and structure analysis, supporting workflows for molecular biology, genomics, and structural biology. Our software is known to be highly accurate as well as easy to use. We also design our software with the end user in mind, and that means modest technical requirements, technical support and documentation, as well as integrated solutions that don't require bioinformatics expertise. This combination of attributes makes our software trusted by users around the world, and we're cited in thousands of peer-reviewed publications each year, totaling over 80,000 as of this year. So our molecular biology applications include tools for DNA and RNA sequence analysis, as well as protein sequence analysis. And this can be anything from basic sequence editing, cloning and primer design, annotation, multiple sequence alignments, as well as Sanger sequence assembly. Our genomics tools are built on our molecular biology and sequence analysis tools and provide additional capacity for NGS analysis, as well as long-read sequence assembly and analysis. The software supports both de novo and reference-guided workflows. It also supports variant analysis and gene expression analysis. Our variant detection options include integrated solutions for variant annotation, and our variant caller is highly accurate, with over 99.7% accuracy in recalling known SNPs. Our structural biology tools, again, building on the genomics and molecular biology applications, are comprehensive and integrate sequence data with structure data. This allows you to perform biophysical property analysis, secondary structure analysis, using the sequence data and apply those findings to the protein structure. So for example, you can locate an epitope based on sequence data and model that on a protein structure. Our structural biology tools also include capabilities for protein design and mutation modeling, as well as protein structure prediction and modeling. For protein modeling, we currently offer 3 tools: NovaFold, NovaFold Antibody, and NovaDoc. NovaFold is used for protein modeling. It's based on the iTaser algorithm from the University of Michigan. And speaking to the accuracy of this tool, iTaser has been the top-ranked automated server in the CASP competition since 2006. NovaFold and iTaser use a novel algorithm that relies on both threading and ab initio folding, which means that we use template structures from the PDB to generate a model from the protein sequence, and then ab initio folding to resolve any parts of the structure that weren't modeled using the template structures. The results are a list of templates and models and a full report showing you different quality metrics and options for downstream analysis, things like protein structure alignment, sequence analysis, and more. We've also made customizations to NovaFold, which allow for custom pipelines for protein modeling. We have a nice case study on our website that shows modeling a GPCR structure using the standard NovaFold approach, as well as a custom library, and combining those to create a hybrid model that was better resolved than either method alone. NovaFold Antibody is a homology modeler, and it uses antibody structures for templates. To demonstrate the accuracy of this tool, we looked at 11 targets from the last antibody modeling assessment, and NovaFold Antibody led the competition with the lowest RMSD value for H3 loop modeling. And NovaDoc is used to model protein-protein interactions. NovaDoc's based on SwarmDoc from the Francis Crick Institute. And SwarmDoc won first place in the Capri competition, that stands for Critical Assessment of Prediction of Interactions. And in this competition, teams from around the world test their tools in a blind study looking at unpublished protein-protein complexes. So SwarmDoc uses many models. It's also unique in that it allows for motion during modeling, creating highly accurate models as a result. Now, in addition to our NOVA tools, we also offer protein design. And our protein design tools are fast and accurate. We looked at our hotspot scanning in comparison to Foldex, Pop Music, and 3 different Rosetta tools, and found that we had the most accurate predictions and the tightest tolerances. In addition, our modeling is incredibly fast. Looking at a Foldex benchmark dataset, our software was able to model all variants on 664 proteins in under 10 minutes. And it's important to note that these tools can be used in exciting ways together. So, for example, our genomics tools can be combined with our protein tools to model variants on a protein structure. Additionally, our molecular biology tools can be paired with our protein tools to guide site-directed mutagenesis for your PCR primer design. And we're currently working with CROs and biopharma companies, combining our protein and genomics tools in different ways, ultimately to support accelerating your pipelines. So things like accelerating phage display or affinity maturation, or building in silico pipelines into your own drug discovery technologies. And already with our partners, we've seen over a 50% reduction in experimental time and costs due to the addition of our in silico tools, which allow for screening of the most promising candidates. So I'd like to share with you some additional resources on how you can get more information. But before I do that, I'd like to briefly hand it over to my colleague Thomas Leary, who's going to show you a quick demo of the software in action using the SARS-CoV-2 spike protein as an example.
Thank you, Katie. Also, I wanted to thank everyone for watching this video and for being part of Bio with us. We're happy to be here. Today, I want to show you some of the most powerful capabilities, in my view, that Protean 3D and NovaFold bring to the table. To demonstrate this, I want to tell a little story about our experience with the COVID-19 crisis. I have loaded up the 6VSB model of the spike protein of the SARS-2 COVID-19. This model was deposited on 2/26 of this year and represented the first model that I saw, or any of us saw, of the spike protein. If we look closely at it, you can see that there's some missing regions on the sides here. Now, these frayed regions are missing atomic coordinate information from the cryo-EM scan. You can also see that over to the right here. There's these grayed out regions. These are regions that just weren't able to pick up the atom structure, possibly due to flexibility, and there was no additional loop there. So it just looks a little jagged and frayed on those edges. Now, we wanted to see, at the time, how well we could help predict these regions using some models that we had actually made about a week before this model was released. And so if I go to my Nova Cloud Services pane here, this is where I can download files that I've submitted via our NovaFold prediction system that's available under this modeling tab here. If you can see around this time, around 220 and 218, we started submitting predictions via our system to help see if we could help make a protein model using NovaFold. I loaded that model into this structure here so you could view it in comparison to the 6VSB model that had the missing regions. So shown in red here, you can see that those regions were reconstituted using the modeling system. Now, if I zoom in closely, in red you can see the model that NovaFold produced in comparison with the model of the cryo-EM structure that was found at 3.5 angstroms deposited on 226. The model that we had produced was found on 220 and 218, in fact. So we had about a 10-day lead time on this molecule. Now, it was at that time that we had an internal discussion that we decided that anybody who needed access to the laser gene tools, including NovaFold, would be granted a 90-day temporary license so they could work from home. Now, this was not an issue at the time, but we started this early so that people could get access to the tools that they might need while their universities across the world were shut down or their research institutes' doors were shut. And we're proud of this, and we wanted to point this out today. Now, in retrospect, we didn't know how good this model was at the time, because all we knew is that we were able to reconstitute the loop regions of the cryo-EM structure, and that's all the information we had. But as the months progressed, more models started coming online. And here I have aligned using our TM-align method one of the models that was released later in March, about 30 days after the model that we had produced. And I want to show you that we had an extremely good correlation between the RBD, which is the receptor binding domain to the ACE2 protein. We had an extremely good correlation in terms of TM score and RMSD. And if I can show you that here in the lower right pane, we can see that the RMSD on this comparison was really low. It was around 1.5 angstroms, in fact. Now, this was exciting for us because at the time, we didn't know how well we did with this model, but we knew that it was important for people to have access to this tool during this crisis. So we released free predictions with every one of those temporary licenses. And now in retrospect, we're really happy we did so, because when we look back at the accuracy of this model and helping to predict this RBD domain, we feel like that we did an extremely good job using this system. Now, I want to thank you guys again for watching this video, and also I want to offer you this 90-day at-home work-at-home license. If you guys need access to these types of tools, please contact me or Katie, and we'll set you up with one. Additionally, we'd like to provide training during this period. I've given many webinars during this crisis, and it's provided a really nice way for me to connect with people all over the world. And I'd like to continue that. So if you feel like having a webinar and receiving a 90-day license, we'd be happy to issue it. Just get a hold of us. But once again, thanks for joining us, and I hope you guys have a really good rest of your bio session.
Thank you, Thomas, and thank you everyone for watching today. Before we end the video, I'd just like to share one website with you, DNASTAR.com/bio2020. If you visit that page, you'll find a link to this recording, as well as resources that you can download, such as the case studies and benchmarks I showed, and a form where you can request access to that free trial that Thomas mentioned. And you can also request a one-on-one demonstration. We'd be happy to put together a custom presentation for you and your colleagues. So we hope to see you again soon, and enjoy the rest of your bio conference.